Welcome back to series of lectures in endocrinology. Today we'll continue with lecture 4 of endocrinology, where we'll get to discuss the hormones that are being released by the posterior pituitary gland. We know to say their production is taking place within the two nuclei in the hypothalamus, and then they'll be transported by axoplasmic transportation to be stored within the axon terminal. So there's neurons that are projecting to the posterior pituitary gland. So after they've been produced by the paraventricular nucleus and the supraoptic nucleus, they'll be transported by the axoplasmic transportation to be stored within the vesicles, just waiting for the signal for exocytosis. So there's a stimulus that will cause exocytosis. So there's two neurohormones to be secreted into the circulation and then to be transported to the target cells where they will have their effect. Okay. So like I said, we are discussing the posterior pituitary hormones. So there are two hormones there, ADH and oxytocin. So the ADH sometimes is referred to as vasopressin because it can have an effect on the blood vessels depending on the receptors it's attaching to. It can attach to V1 receptors of the cardiovascular system. So those smooth muscle cells of the cardiovascular system, they have V1 receptors. Then we have V2 receptors in the kidneys. So depending on which receptors it's attaching to, it has got two different names, ADH and vasopressin. Okay. So the first hormone is antidiuretic hormone, or ADH, which is also called vasopressin. It controls the rate of water excretion in the urine. So how much water is excreted in the urine is controlled by ADH, antidiuretic hormone. So from the name itself, it means it's going to bind to ADH receptors. The ADH receptors are the V2 receptors. So if it goes and binds to the V2 receptors, then it will activate the G protein coupled receptors. Once the G protein coupled receptors are activated, they can go and activate other enzymes like adenyl cyclase enzyme. And when adenyl cyclase enzyme is activated, we all know to say it's going to convert ATP into cyclic AMP, and the cyclic AMP can activate protein kinase A. Once protein kinase A has been activated, it will facilitate translocation of aquaporins 2 to be embedded within the apical side of the epithelium cells. That will now allow water molecules to move from the tubular fluid that is moving within the kidney nephron to enter the epithelium cells, from the epithelium cells to go into the interstitium, and then there will be overall reabsorption of water. So that's the ADH. But when it goes and binds to V1 receptors within the smooth muscle cells in the walls of the blood vessels, it will cause contraction of those smooth muscle cells. So it will bring about general vasoconstriction. So it has got two functions there. So there are two hormones. The other one is oxytocin. And we know to say oxytocin is going to help express milk from the glands of the breasts to the nipples during suckling, and also it helps in the delivery of the baby at the end of gestation. So at the end of pregnancy, gestation, you find that the oxytocin can go and bind to the smooth muscle cells of the uterus, which is the endometrium. So they can go and bind to the endometrium. Those are smooth muscle cells. Then they can contract to help push now the baby during delivery. So it will push now, it will squeeze the baby out of the birth canal. So it's also important when it comes to delivery of the baby. The pars intermedia, of which I discussed in the previous lecture, that the pars intermedia is going to produce melanocyte stimulating hormone, which controls melanin production and secretion for the melanocytes. The melanocytes are cells that will produce melanin, but for them to produce melanin, they need to be stimulated by the melanocyte stimulating hormone, the MSH. So the MSH is produced by the pars intermedia. So there are cells in the pars intermedia in between the anterior and the posterior pituitary gland. There are cells there that are responsible to produce melanocyte stimulating hormone that will go and stimulate the melanocytes to produce and release melanin so that we have skin color. So the melanin that is giving you the skin color is due to the production of melanin by the melanocytes. That needs to be stimulated by the melanocytes stimulating hormone. This is the anatomical structure of the hypothalamus and the pituitary gland, but in this slide we're interested with the posterior pituitary gland. So we say that there are neurons whose cell body are located within the two nuclei in the hypothalamus. So we have these two nuclei in the hypothalamus. The supraoptic nucleus, it means just above the optic chiasma. That's why it's called the supraoptic nucleus. Then we also have the paraventricular nucleus. The paraventricular nucleus is on the lateral side of the ventricles of the brain, where you find the production of cerebrospinal fluid. So we have these two nuclei. So you can see that there are cell or neurons whose cell body are found within these nuclei. Then they are projecting to the posterior pituitary gland. So you can appreciate a lot of axon terminals of these neurons. So once the two hormones are being produced, ADH and oxytocin, they will be transported along the axons, and then they will be stored within the axon terminals here. So you can see the axon terminals where they will store the hormones, the two hormones. So they'll be just waiting for the stimulus for release of these hormones. So this is the structure. So the axon of these neurons, they are actually passing through the infundibulum, the connecting stalk that is connecting the pituitary gland with the hypothalamus. Then we also have the hypothalamic hypophysial tract, which are a band of these axons that are projecting to the posterior pituitary gland. So it's a derivative of the diencephalon. So these are the neurons. The anterior, we said we have a lot of glandular cells. That's why it's called adenohypophysis, because it's derived from the primitive oral cavity. Then you know to say that the hypothalamic neurons, they are the ones that are producing or synthesize oxytocin and ADH, which we know. Then the oxytocin and ADH are transported along the hypothalamic hypophysial tract to the posterior pituitary gland to be stored there. Oxytocin and ADH are stored in the axon terminals in the posterior pituitary gland. So it doesn't mean that this ADH and oxytocin are produced within the posterior pituitary gland. No, they are not produced there. They are just stored there within the posterior pituitary gland. The production is taking place in the hypothalamus, in the two nuclei. Then the oxytocin and the ADH are released into the blood when hypothalamic neurons fire. When these hypothalamic neurons fire, the two neurons that are projecting from the supraoptic and the paraventricular nucleus, when they fire an action potential, it will be propagated towards the axon terminal. That will cause influx of calcium, and that calcium will facilitate exocytosis of the two hormones, oxytocin and ADH. A bit on the chemical structure of ADH and oxytocin. So both oxytocin and ADH vasopressin are polypeptides. So we said for them to be polypeptides, they need to have a sequence of amino acids that will be less than 100 amino acids. So those peptide chains, they should contain less than 100 amino acids for them to be polypeptides. So they qualify to be peptides. They are not hormones. So they are not protein hormones, but they are polypeptide hormones because they have a sequence of less than 100 amino acids. So each containing 9 amino acids. So in their structure, they have 9 amino acids, vasopressin and oxytocin. So if you count these, when you count these amino acids, you know to say that there are 9 amino acids. But those of interest are the amino acids, the phenylalanine amino acids. So you can see we have the phenylalanine amino acids and arginine. So the phenylalanine amino acids is amino acids number 3 in the sequence, and the arginine is amino acid number 8 in the sequence. So in this sequence of amino acids, the structure of vasopressin and oxytocin is similar. The only difference is the phenylalanine and arginine on the amino acids number 3 and 8 on the sequence. So in oxytocin, instead of finding the phenylalanine, you're going to find isoleucine amino acids. So on sequence number 3, instead of finding phenylalanine, we're going to find isoleucine. Then on sequence number 8, there is leucine amino acids. So this is the difference in structure. So note that these two hormones are almost identical in terms of structure, except that in vasopressin, the phenylalanine and arginine replace the isoleucine and leucine of the oxytocin molecule. So this is the only difference. So because of these similarities in structure, they will also result into having partial functional similarities in terms of function. So the receptors to which oxytocin can bind, to some extent even vasopressin can also bind. Why? It's because these two hormones are almost identical in structure. But because of the same minor differences, you find that they'll have different effects on these receptors when they go and bind. So vasopressin will bind to different receptors, and oxytocin will also bind to different receptors. So the similarity of the molecules explain their partial functional similarities. So in terms of function, they have a partial functional similarities because of their structure. They are similar in structure. So I hope you get that. Okay, so you can see in this diagram, this is oxytocin. This is more of biochemistry. So you don't really need to remember all these structures, but what you need to remember are actually the similarities. You can just remember the similarities. You don't need to know how to draw this structure, okay, because this is physiology, it's not biochemistry. So the oxytocin and ADH, you can see from this diagram that the structure is almost the same, but on amino acid number 3 and 8 we have differences. In oxytocin, on amino acid number 8 and number 9, number 8 and number 3, we have leucine and isoleucine. But in that of ADH, we have phenylalanine and arginine. That's why the other name for vasopressin is referred to as arginine vasopressin, because of this arginine that is found in the structure of vasopressin. So it's called ADH or vasopressin, dependent on the receptors it's attaching to, whether it's V1 receptor or V2 receptor. V1 receptor in the cardiovascular system is referred to as vasopressin. If it goes and binds to V2 receptors in the epithelium of the kidney nephron, then it's referred to as ADH, antidiuretic hormone. So what are the physiological functions of antidiuretic hormone? The physiological functions of ADH, antidiuretic hormone. ADH increases the permeability of the collecting ducts and tubules to water and allows most of the water to be reabsorbed as the tubular fluid passes through these ducts, thereby conserving water in the body and producing very concentrated urine. So you know to say ADH, when it goes and binds to ADH receptors, the V2 receptors, I said it's going to activate the G protein, and that G protein will activate enzymes like adenylyl cyclase enzyme and phospholipase C. We all know that. The adenylyl cyclase enzyme, it will convert ATP into cyclic AMP. The cyclic AMP will go and activate the protein kinase A. The protein kinase A now will cause phosphorylation, not phosphorylation, but it will cause translocation of the vesicles that contains aquaporins 2. So the aquaporins 2 now will be embedded within the apical side of the epithelium cells that will allow movement of water. So overall, we have reabsorption of water. So you are not excreting a lot of fluids in the urine. You'll be producing concentrated urine, but you are conserving water in the body. So there is conserving water in the body because of you are reabsorbing much of the water. You are not excreting much of it because of ADH. But in the absence of ADH, the collecting tubules and ducts become almost impermeable to water, which prevents significant reabsorption of water and thereby or therefore allows extreme loss of water into the urine, also causing extreme dilution of the urine. So now in the absence of ADH, it means that the collecting tubules and the collecting ducts, they will be impermeable to water because they will be lacking the aquaporins too. So there will be no reabsorption of water. If there is no reabsorption of water, much of the water will be excreted in the urine. So you'll be producing diluted urine. Urine will be diluted and the quantity of the urine will be very high, so the quantities will increase. The quantities of urine will increase because now you are not reabsorbing much of the water, so you are producing diluted urine. The diluted urine, because there is much of water that is being excreted with the electrolytes, so they won't be highly concentrated because of the presence of water molecules that will be diluting them. So the higher concentrations of ADH have a potent effect of constricting the arterioles throughout the body and therefore increasing the arterial pressure. For this reason, ADH has another name which is called vasopressin. So in higher concentration of ADH, it doesn't only have an effect on the kidney nephron, but it can go and bind to V1 receptors within the cardiovascular system that will bring about vasoconstriction. So if the blood vessels are constricting, you know to say peripheral pressure or resistance is going to increase, and that will cause an increase in blood pressure. So an increase in secretion of ADH, there will be an increase in the concentration of ADH, then that can have a potent effect of constricting the arterioles. So you find that all the arterioles will be constricting, increasing the peripheral resistance. And if the peripheral resistance is increasing, we all know to say that the blood pressure is also going to increase. That relationship was explained in the Poiseuille-Hagen formula in cardiovascular system. So I hope you still remember the relationship there. So this is what I was trying to explain, to say in the blood vessels, it can go and bind to V1 receptor, but in the kidney nephron, it will go and bind to V2 receptor. So if it binds to V1 receptor, it will increase systemic vascular resistance. That will cause an increase in arterial pressure. But if it goes and binds to V2 receptor, it will cause an increase in reabsorption of water. So the blood volume is going to increase as well. Once you have an increase in the blood volume, there will be an increase in arterial pressure as well. So the blood volume, if it's increasing, the blood pressure is also going to increase because the plasma volume is increasing. That will have an effect on arterial blood pressure as well. This is the actual mechanism of ADH on the kidney nephron, of which I've already explained. So the ADH will be secreted, especially if the concentration of solutes in the extracellular fluid is increasing. So if the osmolality in the extracellular fluid is increasing, the osmoreceptors in the hypothalamus, they are going to respond by stimulating action potentials in those two neurons that are originating from the paraventricular nucleus and the supraoptic nucleus. Those action potentials will be propagated down the axon to the axon terminals. That will bring about opening of calcium-gated ion channels. Influx of calcium will cause exocytosis. So the two hormones now, remember the hormones after being produced, they will be stored within the axon terminals in form of vesicles, waiting for the signal for exocytosis. So this signal now is the action potential. So if the osmoreceptors are responding to osmolality, if there is high osmolality, then they are going to initiate action potentials within those neurons. That will be propagated down the axon terminals. Then they will stimulate the calcium-gated ion channels within the axon terminals. The influx of calcium is very necessary for exocytosis of ADH and oxytocin. So ADH and oxytocin will be released. So in this case, we're interested in ADH that is being released. And you know to say ADH is a polypeptide. So it's more like a protein in nature. It's a polypeptide. It's a combination of amino acids, a sequence of amino acids, 9 amino acids. So because of that, it's water soluble. It doesn't need to bind to plasma protein for it to be transported. So it will just dissolve in plasma, then it will be transported by the cardiovascular system to the kidneys. In the kidneys, it will leave the capillaries to enter the interstitium, and then it will go and bind to receptors that are on the plasma membrane of these epithelium cells. So they will go and bind to G protein coupled receptor. So you can see the G protein coupled receptor that are being activated once ADH binds there. Then the alpha protein subunit will dissociate itself from beta gamma protein subunit. The alpha protein subunit can go and activate the adenyl cyclase enzyme or adenylate cyclase. Adenyl cyclase enzyme will be activated, and then it will convert ATP into cyclic AMP. So adenosine triphosphate into cyclic adenosine monophosphate. So the cyclic adenosine monophosphate is going to activate the protein kinase A, meaning that the activation of the protein kinase A is dependent on the cyclic AMP. So it's cyclic AMP dependent protein kinase. In the presence of cyclic AMP, it will be activated. Then it will cause translocation of these vesicles. So you can see these vesicles, they are containing aquaporins too. But if they are within the cells, in the cytoplasm of the cells, this cell won't be permeable to water. So for it to be permeable to water, you need to translocate the aquaporins too, that will now be embedded within the apical side of the epithelium cells. And when that happens, water can move from the tubular fluid. So you can see the tubular fluid that is moving in the nephron here. Now it can be reabsorbed. It will enter across the aquaporins too. The aquaporins too are protein channels for water molecules. So it will allow movement of water molecules to enter the epithelium cells. Then on the basolateral side of the membranes, we have the aquaporins 3 that can also allow movement of water molecules. Then these water molecules will move into the interstitium, it will cross the cardiovascular wall because you have the capillaries, and then the capillaries, they have those pores that can allow movement of water molecules. So there is overall reabsorption of water molecules. So the mechanism here, you need an increase in cyclic AMP in the cells and the formation of microtubules in the cell membrane. So these microtubules, they could be used for transportation of the aquaporins too. So you can see the transportation of aquaporins too to the apical side of the membrane. That will allow reabsorption of water. So I said the collecting tubules and collecting ducts under normal circumstances, they are impermeable to water. As long as you don't have ADH, they don't have the aquaporins too, so they are not going to be involved in the reabsorption of water. But in the presence of ADH via the V2 receptors, then you can see the appearances of aquaporins too that will facilitate the reabsorption of water, just like I've already explained. Okay, so that's the mechanism for ADH. How is it regulated? So regulation of ADH secretion. So you don't want to be producing ADH even when the blood pressure is increased, even when you have high water content. If you have an increase in blood volume, do you still need ADH? No. So what is going to be regulating the secretion of ADH? The ADH secretion is regulated by osmoreceptors in the hypothalamus, like I explained. So these osmoreceptors, they are sensitive to the osmolality or the extracellular fluid compartment. So if the extracellular fluid compartment is becoming hyperosmolar, then these osmoreceptors, they are going to respond by triggering action potentials in those neurons in the paraventricular nucleus and supraoptic nucleus. Concentrated body fluids stimulate ADH secretion. So the concentrated body fluid, they are going to stimulate ADH secretion because if the body fluids are concentrated, it means that the osmolality has increased. The concentration of these dissolved substances in solution has increased for them to have a concentrated body fluid that is going to stimulate ADH secretion. Whereas dilute body fluid is going to inhibit. So if the extracellular fluid compartment is hyperosmolar, meaning that it's reduced. Hyperosmolar is going to inhibit the secretion of ADH. In other way, if the electrolytes are highly diluted in the extracellular fluid compartment, it means you have a lot of water. So you find that the secretion of ADH is going to be inhibited. ADH is also under cardiovascular regulation because it has got an effect on the blood pressure, the arterial blood pressure. We said an increase in the concentration of ADH is going to cause vasoconstriction. That will increase the arterial resistance, causing an increase in blood pressure. So it's also regulated by the cardiovascular system itself. So decreased blood volume is going to stimulate ADH secretion. So if the blood volume or plasma volume is reducing, it means blood pressure is reducing. So that is going to enhance the secretion of ADH. That will in turn try to compensate that by causing an increase in blood pressure. So you find a decrease in blood volume, it's going to stimulate ADH secretion, which makes sense. Decreased stretch of the baroreceptors or the carotid, aortic, and pulmonary regions also stimulate ADH secretion. The baroreceptors are the ones that are responding to pressure changes. So if there is a decrease, a stretch on the baroreceptors, it means that the arterial pressure is reducing to some extent, because an increase in the arterial pressure is what is going to be stretching the baroreceptors. So if the baroreceptors are not being stretched more, it simply means that the blood pressure is reduced and the blood volume is also reduced. So that in itself can also stimulate the secretion of ADH from the posterior pituitary gland. So here is the posterior pituitary gland and stimulatory stimuli that can cause an increase in the secretion of ADH. So the first one there is angiotensin 2. The angiotensin 2, of course, is going to stimulate the release of ADH. Why? It's because angiotensin 2 is going to be secreted when there is reduced blood volume or reduced blood pressure. Remember the renin-angiotensin-aldosterone system. So once the renin is being produced by the kidneys, when there is low blood pressure or when there is low blood volume, we say that the renin is going to convert a precursor that is being produced by the liver, the hepatocytes. The hepatocytes, they are producing angiotensinogen. The angiotensinogen will be converted into angiotensin 1. Then the angiotensin 1, after it has been converted by renin, it will now be transported to the lungs and to the kidneys, where you have angiotensin converting enzymes that are going to convert angiotensin 1 into angiotensin 2. The angiotensin 2 has got a lot of vascular function. So one of the functions, it can go to the posterior pituitary gland to stimulate the release of ADH. So under the influence of angiotensin 2, the secretion of ADH is going to increase. And of course, I told you to say the angiotensin 2 is also acting via the phospholipase C pathway, via the activation of G protein-coupled receptors that will increase calcium levels, and then that calcium levels will encourage exocytosis of ADH. Sympathetic stimulation, of course, is also going to stimulate the release of ADH. Then the hyperosmolality, if there is hyperosmolality, you know to say that the extracellular fluid is highly concentrated, then that can stimulate the osmoreceptors, so the secretion of ADH is going to increase. Hypovolemia, if there is low blood volume or low plasma volume, that can also enhance the release of ADH. Hypotension, low blood pressure in itself, we say that the baroreceptors will be stretched less. So if there is decrease in the stretch of the baroreceptors, there is a neural pathway that will go and stimulate the release of ADH. Then once you have this ADH, which is also called vasopressin, so vasopressin, when it's binding to V1 receptors in the cardiovascular, that will bring about vasoconstriction, increasing the arterial resistance. The arterial resistance, if it's increasing, there will be an increase in arterial pressure, blood pressure, which you can see here. So it's a regulatory mechanism for blood pressure, so that the blood pressure is regulated, is maintained within the acceptable range of the blood pressure, the systolic blood pressure and the diastolic blood pressure. Then on the other hand, it has got an effect on the kidneys via the V2 receptors. It's called antidiuretic hormone. So it's going to increase reabsorption of fluids from the kidney nephron. And then the blood volume is going to increase, because if you are reabsorbing a lot of water, that will add on to the volume of plasma or blood. So the blood volume is increasing. That in itself can also cause an increase in arterial blood pressure. The physiological function of oxytocin. So to some extent, we've already discussed this, but just adding more information for students to appreciate more. The physiological functions of oxytocin. Oxytocin causes contraction of the pregnant uterus, especially towards the end of gestation. So towards the end of gestation, there is development of the endometrium. Those are the smooth muscle cells. They have oxytocin receptors, which are G protein-coupled receptors that can cause contraction. So you know to say oxytocin is going to be released during labor, and that will help in the delivery of the baby during giving birth. Oxytocin also help in milk ejection by the breast. So it's going to aid in milk ejection by the breast, because within the breast we have the myoepithelium, and those myoepithelium, they have receptors for oxytocin. So once oxytocin goes and binds to those myoepithelium, there are also G protein coupled receptor that will cause contraction of the myoepithelium to squeeze the milk out of the ducts. From the acina ducts, the acina cells are the ones that are producing the milk, and then to be secreted into the duct system. Then if the myoepithelium, they are contracting, they are squeezing the duct system to express the milk via the nipples. So the suckling stimulus on the nipple or the breast causes signals to be transmitted through the sensory nerves to the oxytocin neurons in the hypothalamus, which causes release of oxytocin by the posterior pituitary gland. So if the baby is suckling on the nipples, within the nipples you have receptors that are sensitive to touch. So when the lips of the baby are touching the nipples, they are going to stimulate those receptors, and then they will generate an action potential via the sensory nerve fiber. It will project to the hypothalamus. So in the hypothalamus, it will go and stimulate those neurons that are producing oxytocin. Then they will fire an action potential again from there, from the supraoptic and paraventricular nucleus. Then the action potentials will be propagated towards the axon terminals that will cause influx of calcium and exocytosis of oxytocin. The exocytosis of oxytocin is facilitated by the presence of calcium, because we said the calcium will go and bind to V-snare, T-snare interaction, synaptobrevic syntaxin interaction will bring about exocytosis of the oxytocin. The oxytocin now will be transported by the cardiovascular system to the myoepithelium in the breasts. So in the myoepithelium or the breast we have oxytocin receptors, and those will cause now contraction. Then milk will be expressed via the nipples or the breasts. That's what is going down there. The oxytocin is then carried by the blood to the breast, where it causes contraction of myoepithelium cells that lie outside of the form of a lattice work surrounding the alveoli of the mammillary glands. So the mammillary glands, these are the breast where milk is being produced. So if these myoepithelium are contracting, then they are squeezing the milk out of the breasts. So in less than a minute after the beginning of suckling, milk begins to flow. So just less than a minute, because the oxytocin will be now transported to the myoepithelium. Now what you need to know here is, as long as the nipples are stimulated, the neurons there, the sensory neurons and the receptors, they can't differentiate between the baby and the father or the boyfriend. So as long as they are stimulated, then milk will start dropping down. Why? It's because of oxytocin. So even if it's a man licking the nipples, if it's a boyfriend licking the nipples, or whatever is licking the nipples, that can stimulate the production in the secretion of milk from the breasts. The mechanism is the same. This mechanism is called milk letdown or milk ejection. So milk letdown or milk ejection, facilitated by the presence of oxytocin. So this is the pathway. The stimulus that is taking place is the suckling at the nipple. So there are sensory receptors that will be stimulated, the tactile receptors or touch receptors. Once they are stimulated, they are going to generate an action potential via the sensory nerve fiber. It will project to the hypothalamus. Then in the hypothalamus, it will be stimulating those neurons that are producing oxytocin in the supraoptic and paraventricular nucleus. So you can see this sensory information is an action potential. They need to stimulate this fiber to fire an action potential. Then another action potential will initiate here. Then it will be transmitted down to the axon terminals. There it will bring about influx of calcium, exocytosis of oxytocin that will be transported by the cardiovascular system or the blood vessels, the bloodstream, to the breast to go and stimulate the contraction of myoepithelium, squeezing the milk out of the nipples. Then the baby will benefit from the milk that will be ejected out of, or the milk that will be expressed out of the nipples. On the other hand, we have the oxytocin that will have an effect on the endometrium, so the smooth muscle cells of the uterus. So during childbirth, what happens is the baby will start positioning itself into the birth canal, stimulating or stretching the cervix during late pregnancy. So during late pregnancy, if the cervix is stimulated or stretched by the movement of the baby, that will cause an action potential, because there are sensory receptors that are also sensitive to stretching. So they are mechanosensitive receptors that are connected to a nerve fiber, sensory nerve fiber. So an action potential will be initiated here. It will be propagated via the sensory nerve fiber to the hypothalamus and stimulating this nerve neuron, or the nerve that is producing acetylcholine, to cause another action potential, influx of calcium, exocytosis of oxytocin, and that oxytocin will be transported by the bloodstream to the uterus. Then it will go and bind to smooth muscle cells of the uterus to cause contraction, squeezing the baby out of the birth canal. So this is labor that is taking place. So there are strong contractions that is due to oxytocin. So if a woman is not producing strong contractions, sometimes labor can be induced with oxytocin. So you can just inject oxytocin, or administration of oxytocin will bring about the same effect, because it will go and bind to these smooth muscle cells, then it causes contraction. So that will result into labor, which is called induced labor. Okay, but the mechanism or the physiology should be the same. So this is the mechanism of oxytocin action. How does it work? So you have G protein-coupled receptors on these smooth muscle cells of the endometrium. It's responding to oxytocin. So oxytocin comes and binds to the G protein-coupled receptor, activating the G protein. The alpha protein subunits can dissociate itself from the beta-gamma protein subunits. Then it can go and activate the phospholipase C. Once the phospholipase C is activated, we all know to say it's going to convert phosphatidylinositol triphosphates or bisphosphate into DAG and IP3. So you can see IP3. So phosphatidylinositol bisphosphate and it will be converted into DAG and IP3. And you know to say DAG is fat soluble, so it will be moving within the cell membrane of these cells, but the IP3 is water soluble. It will dissolve in the cytoplasm. It will go and bind to the IP3 receptors of the sarcoplasmic reticulum. So these are ligand-gated ion channels. So when IP3 goes and bind, they are going to open to allow movement of calcium. So it's a channel that is going to open to allow movement of calcium. Calcium will go and bind to camodulin. Calcium camodulin complex will go and activate the myosin light chain kinase. It's a kinase. What does it do? Phosphorylation reactions. So it will phosphorylate the myosin heads, and once the myosin heads in smooth muscle cells are phosphorylated, they are going to interact with actin. Actin-myosin interaction brings about smooth muscle contraction. So there will be strong contractions of the uterus because of this interaction of the hormones and the receptor that will trigger a cascade of reactions within the smooth muscle cell. Pineal gland, there is nothing much about the pineal gland, but we know to say it's the gland that is producing melatonin, and melatonin is a derivative of tryptophan amino acid. I think you remember that. So there are some functions of this gland which are not well understood. Maybe with time there will be a lot of discoveries by scientists, and then we'll understand the function of the pineal gland. But what we know so far, the pineal gland, it is known from comparative anatomy that the pineal gland is a vestigial remnant of what was a third eye located high in the back of the head in some lower animals. So in some lower animals, it was believed that the pineal gland was supposed to be the third eye at the back here, higher in the neck region, or the head itself. But in human beings, we don't have the third eye, so it's a vestigial remnant that can produce some hormones, melatonin hormone. So some of the functions that are not well understood, the functions of the pineal gland due to the hormones that it's producing there, it has been described as the seat of the soul. The seat of the soul, so it has got something to do with the soul of a human body or a human being. Then it's going to enhance sex, so it's also involved in enhancing sex pleasure during intercourse or sexual intercourse. So it's going to enhance sex, mechanism which are also not well understood, because the melatonin that can also stimulate certain centers that are involved in the pleasure when you are having sex. So they are going to enhance sex, staving off of infection. So staving off of infection, promoting sleep, because you have melatonin that is involved in the circadian rhythm. So melatonin is involved in the circadian rhythm, so that you can be awake at one point, at another point you feel like sleeping. So it's more like the clock of the body, the natural clock of the body. Even when you wake up, you can guess to say the time should be around 7 hours or 6 hours. How do you guess that? It's because of this pineal gland that is producing melatonin. Okay, so that melatonin is more like the clock of the body. Then you're able to tell time of the day, especially in the morning. Then it's also there to enhance mood. So it's enhancing mood, good moods, bad moods. Those can be enhanced by the pineal gland, melatonin hormone that is being produced there. So there is modification of certain neurotransmitters that are involved in mood, increasing longevity of moods. So even longevity of moods, for you to be happy for a long period of time, maybe that can be enhanced by the pineal gland, because it's producing the melatonin hormone. So the pineal gland is just behind the epithalamus. So it's on the posterior aspect of the epithalamus. So you can see this tiny red structure here. This is the pineal gland. That's why I say it to be a vestigial remnant in human beings, because it's very small, just at the back or the posterior aspect of the epithalamus. So the pineal gland, or the pineal body, is a small cone-shaped structure attached to the posterior region of the epithalamus. So like I said, it's attaching to the posterior region of the epithalamus. It's producing or secreting melatonin that helps regulate circadian rhythm, the 24-hour body clock. So you're able to tell the time of the day because of the melatonin that is being produced. It also appears to affect the synthesis or the production of hypothalamic regulatory hormone responsible for follicle-stimulating hormone and luteinizing hormone. So this regulatory hormone actually is called the gonadotropin-releasing hormone that is being produced by the hypothalamus. So the production of gonadotropin-releasing hormone is affected by melatonin. So the pineal gland, it has got an effect on the production of gonadotropin-releasing hormone. Maybe that's why it has got an effect on the pleasure of sex or emotions, because it has got an effect on these hormones, which are gonadotropins, the follicle stimulating hormone and luteinizing hormone, indirectly by regulating the production of gonadotropin releasing hormone. Then it has got a function in sexual maturation, which is also not well understood. So at your own time, you need to find out more about this pineal gland, but the most important hormone there is the melatonin and how it comes into circadian rhythm. Okay, so we're not expanding much here because there will be a lecture dedicated to physiology of sleep. That's where we're going to discuss in detail the melatonin and the waves that are produced by the brain itself when you are sleeping, when you are awake. So there are certain waves that are being produced. All that will be discussed under the physiology of sleep. Otherwise, we are done with this lecture. Okay, so I will see you in another lecture. For now, we are done. This is where we're going to end. Thank you very much for your attention.